Testing, 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 testing. 1, 2, 1, 2. Testing.
And the Q&A mic is good too.
Tyler, they're about to test the Q&A mic for you.
Oh yeah, I see on Commons View.
Something, whatever. Testing,
Hello?
testing, testing,
Testing, testing.
testing, testing. 1, 2, 3. Testing, testing, testing.
Hello,
1, 2,
hello,
3.
there. Hello. Hello.
Perfect.
Testing.
So the balance there between the two mics was really off. Are they both at different levels right now?
Oh, I was chatting with Tyler. Tyler. So he was just looking away from the monitor and the screen above. So he was facing away from the lapel mic when he was testing because he was watching Merrick. So you want to, yep. Which is why we put it on your left shoulder. So when you look up there, you don't fade out.
Yeah, nicely put. That's a tough placement with his shirt. Is that the laser pointer? The thing from Jim Coyer?
No, that was just his mouse set up in laser pointer mode. Sorry, Tyler.
I was very enthusiastic for a second if it was that easy. I don't know if it is. All right, I'm going to start the presentation.
Let me check with Merrick, see if he's ready.
Okay.
Merrick, Tyler was going to start the presentation of the webinar now. If you wanted to, if you're okay with that, Tyler was going to start the webinar. Go right ahead, Tyler.
Thank you.
He was just talking a minute ago. So I wanted to make sure we were all good.
All right. Hey, good evening. Stragglers, come on in, have a seat. All right. So good evening, everybody. Welcome to another Shoals Marine Lab seminar, our Rock Talks. Really excited to have everyone here tonight. For those of you that I haven't met, I'm Dave Buck. We welcome all of y'all tonight. Our Rock Talks, for those of you that were here last week, you know the drill. For our new arrivals, these are our weekly seminars. It's a chance for our entire campus to come together and listen to a lecture. Really fun opportunity for all of you to sit back and listen to a wonderful talk. These seminars have been going on here at the lab for many, many years. We call them Rock Talks in part because in the early days of the lab, seminars would be given actually out on the rocks around the intertidal. So the format for tonight's talk will be about a 45, 50 minute, could go 3 hours. We're not quite sure yet. It'll be a talk with a period of question and answer at the end. I'm going to be carrying around this microphone. So when you have a question, just raise your hand. I'll come to you with the microphone so that everyone can hear you clearly, so that Andy can hear you clearly, and we'll go about it that way. We'll try to end pretty close to 9 o'clock if we can. So I don't think we have any online presence, so I'm not going to really introduce everybody, although I do want to give a quick shout out to, we have 2 new sort of groups on island. For those of you that have been here for a week, the room is very full. So let's give a welcome to our Marine Science for Teachers class. This is a group of mostly New Hampshire and I believe a couple of Massachusetts educators that are here learning about how to integrate marine sciences into their public school curriculum. Then we also have our intertidal ecology program that got on island yesterday. Very happy to have them here. The intertidal ecology program is, they're contributing to our longest running data set here at the lab. Got started in the late 70s. We lost a couple of years there, and it's now currently from 1982 to present. So we're really happy to have y'all here. So welcome intertidal ecology interns and mentors. So without further ado, tonight's rock talker is Andy Turko. Many of y'all should recognize Andy because he's been here for a week. If you don't recognize him, where you been at mealtime? Dr. Andy Turko is a comparative and evolutionary physiologist. He's just recently been appointed assistant professor at Guelph University. Yeah, shout out, congratulations. And his position at Guelph is sort of a bit of a homecoming because he did his bachelor's, his master's, and his PhD there. All right, sorry. All right, well then I won't say. All right, so I'm not going to talk about any of that. So in his lab at Guelph, his research program investigates how animals integrate physiological and behavioral responses to changing environment. And he first came here in 2015 as a teaching assistant for IMBL. He came back as faculty. This is his 3rd year as faculty. We're really psyched to have Andy. And I'm very happy that he's here because as he and I were discussing earlier, it's time for us to, one of his stated goals at Coming East is to have a tour of local clam shack. So that's why he's here, in addition to giving us a great lecture. So Andy, welcome. That's right. So please join me in welcoming Andy.
Okay, thanks everyone for coming out. It's gonna be fun. I'm gonna reset my timer so I don't talk for 3 hours. So for those of you who know me, you're gonna be bored for the first 60 seconds, but for those of you who don't know me, this is my whirlwind introduction to the island. So who am I? As Dave said, I did my bachelor's degree at the University of Guelph in Ontario, Canada, over here about an hour west of the great city of Toronto. And I went there because they had one of the only freshwater biology programs in the country. And I'm kind of a weirdo. I never really was interested in the ocean nearly as much as I was in the streams and rivers inland. So that's why I went there. I expected I'd be there for 4 years and not a day more. And then 13 years later, I graduated with a PhD from that university, University of Guelph. And so during the 9 years or so, I was a graduate student there. I was really focused on this question of how amphibious fish survive on land. Like many of us, probably, I've always been fascinated by really weird animals, animals that do unexpected things. And I would argue that fish living on land is one of the most unexpected things an animal to do. And so I'm going to talk about that work today. That's still work that I like to do. But after my PhD, I wasn't really sure how my career, my life would unfold. And I really wanted to gain some more applied skills. And I wanted to use some of the physiological toolkit I had learned as a PhD student to actually work on applied conservation programs. So I went to the University of Windsor, right across the river from Detroit. And I started investigating why red-side dace, this really beautiful minnow here that lives in northeastern North America, tried to understand why their populations are declining. Their populations are down like 80% in the last 30 or 40 years. We don't know why. We want to save them. So I was using physiology to try to answer some of those questions. I then sort of carried that program with me 2 years later to McMaster University in Hamilton. And here I joined a behavioral ecology lab to try to gain some new academic skills, try to be a behavioral ecologist, which is really a difficult thing for a physiologist to learn how to do. But I did some experiments trying to understand basically how fish choose where to live. So I used cool tools like shuttle boxes here that track how fish interpret their environment and make decisions about the conditions they want to experience. And as Dave said, a major goal of my lab is trying to integrate this decision-making process with the underlying physiology to understand how animals deal with a variable world, basically. After that, you'll see in some of my slides to come that an underlying theme of my work is understanding how fish breathe and how fish get oxygen from air and water. And that has gotten me really interested in questions of how fish regulate gill structure and function. So that's what I'm currently doing at Wilfrid Laurier University, asking these more mechanistic questions about how fish sort of choose how much surface area they expose to the world. But it's all been given away. The good news, the exciting news is that starting in January, I'm going back to where it all started, back to the University of Guelph, where my research program is hopefully going to explore this question of how animals respond to changing environments. And the plan is to really focus on native minnows. Minnows are amazing fish. And in Ontario, we've got a bunch of species that live in the same streams. Some of them are endangered, almost extinct. Others seem to love human disturbance. And so the early focus of the lab is really going to be trying to understand why some species are so sensitive to environmental change and others are robust. So that's how I'm thinking about environmental change now. But now we're going to go back in time a few years when I was really thinking about a different kind of environmental change. And that's how amphibious fishes cope with this transition between water and land. So I want to play a little game, little survey. How familiar are people here with amphibious fish? Can anyone here name a fish that lives on land? A mud skipper, great example. Snakefish, snakeheads, Shanna, yep, definitely. Lungfish, great example. There are lots of them. So those are all great examples. They all made it on the slide. So I've always been really impressed by the diversity of amphibious fishes. So we have critters like lungfishes that can survive for more than a year without water. They tuck themselves into a little mucus cocoon and they just shut everything down, shut down their metabolism and just wait for rain to come. We have other species like polypterus and mud skippers that live in marshes and mud flats, spend most of their life out of water, and then other species like grunions that are mostly aquatic. They hop onto the beach to lay their eggs and then hop back in the ocean. So this phylogeny here shows all extant fishes. This is at the order level. I don't want you to you don't have to read any of the words. But here it's coded so that blue orders are fully aquatic and bolded black orders are orders that contain amphibious species. When I first plotted this phylogeny, it absolutely blew my mind because there are amphibious fishes all over the place. This seems to be something that just keeps evolving again and again and again in extant fishes, which really surprised me. I thought there were lungfish, there were mud skippers, there were the killifish I'll talk about in a little bit. But this is something that happens again and again. Another game, how many species of amphibious fish might there be in the world? Price is Right rules, can't go over. Any guesses? Dave? 28. 50. 3,700. That's over. How many, who knows how many species of fishes there are in the world? It could be. The number that I learned is 20,000, but that could easily be 30,000 by now. So there are a lot of fishes. There are actually only 200 amphibious ones. So it's a lot compared to guesses of 30, but this is like less than 1% of fishes on Earth. But what's really astounding is that, as I said, this is something that keeps evolving again and again. 200 species of fish, and yet we think the ability to live on land is independently evolved between 40 and 90 times. More recent estimates are at the higher end of that scale. And so, I don't know, it's hard for a fish to live on land. These numbers continually blow my mind. This has happened 90 times in the extant fishes that currently live on Earth. And so I've been really fascinated with how this transition unfolds and how fish survive on land. I think I mentioned in my spiel, though, that I was going to talk about why fish leave water. I'm not going to talk a lot about that, but I'm going to ask you why fish leave water. Why do you think a fish might move onto land? To get food. Yes, amphibious fish move onto land to get food. Escape predators, absolutely, lots of examples of that. Lay eggs on land, yep. They can avoid aquatic predators as adults, but they can also get their eggs out of scary situations. The fungulus and tide pools here do just that. They move onto land, they spawn at the really high end of the high tide, and so their eggs develop in air. Yeah. Less competition, absolutely. Lots of examples. Abiotic factors, the water gets disgusting and they get out of there. Absolutely. A lot of these amphibious fish live in the most horrendous swamps and bogs and places where no fish should ever be found. When it gets really bad, they can get out. The reason I don't want to spend a lot of time talking about why amphibious fish leave water is because I could go all night. Basically, any reason you can think of, there's probably an example. These fish are hyper diverse. All the species have totally different natural histories. They jump out of water for whatever reason you can come up with. But we're going to leave it at that. Why am I interested in amphibious fish? One of the reasons is because I am interested in where we come from as humans. We are all highly derived, really weird amphibious fish. And so if we want to understand where we came from, I would argue that until someone smart invents a time machine, the best thing we can do is we can look at the fishes that are currently experiencing this transition today. And so the first part of my talk, I'm going to ask this question: how does this process unfold? How do fish evolve to become amphibious? This is something that I've been really interested in the last few years. And then we're going to shift gears to a different kind of how question, and I'm going to tell you a little bit about how amphibious fishes survive the challenges of life on land. So I'm a comparative physiologist by training, and so we're going to talk about the physiological adaptations that killifish in particular use. These are the fish that I dedicated most of my PhD time to working on. I'll explain why in a little bit. They're pretty awesome now. So part one, how does amphibiousness evolve? I want you to all think about this a little bit. How do you envision this transition occurring? You have a fish swimming around in the ocean. It has to move on to land. What's the sort of order of operations in evolutionary time? Has anyone thought about how this might happen? Anyone? They need to have lungs, so they gain the ability to breathe air in water, and then that helps them leap onto land. Absolutely. They need to be able to keep their gills wetted. They need to support their bodies on land. There's a lot of gravity on land, absolutely. But if you're thinking about they keep their gills wet, they support their bodies, how does that work? And what are the transitions? What would you expect the transition animal to look like? Like a frog. So a frog breathes air, moves on to land. Okay. So that idea, I think, is by far the most common. I call it the continuum hypothesis because I can't think of a better name for it. If anyone has a better name for it, I'd love to hear it because I don't think this is very catchy. But the continuum hypothesis is basically like the textbook view. You have aquatic animals that they evolve to breathe air because they live in swamps, they live in intertidal areas, they live in places where air breathing is advantageous. They evolve lungs or some sort of air breathing organ. All of a sudden, this major hurdle to colonizing land is gone, and then they can evolve all these interesting terrestrial, amphibious, natural history. How else could this work? So they just get better at moving around. And then they walk onto land and then they solve the problem. Sure, that's basically the alternative. I view it slightly differently, but I think that model works. So the other big model that scientists have considered for a long time is what I call the emergent first hypothesis. And so this is the idea that a water-breathing fish moves onto land, crawls onto land because it has awesomely strong pectoral fins. So we would call that like a terrestrial pull. So we think about why fish leave water. I meant to say this earlier. We think about two main categories. We think about pushes and pulls. The aquatic environment is either harsh, full of predators, or it's full of natural substances, and that pushes fish onto land, or resources, opportunity on land pulls the fish out of water. So this model envisions either a fish that lives in an intertidal habitat that disappears, a seasonal habitat that dries out in the dry season, something happens, or the fish sees an opportunity on land, leaves water, and then air breathing evolves secondarily. I was really curious which of these explains most of the 80 or 90 independent origins of amphibiousness on Earth. I had no idea how to go about this, so I put on my IBM, my integrative marine biology laboratory hat, started thinking about predictions. I actually spent weeks just thinking about what these two hypotheses predict. I'm going to ask you to do that now in 30 seconds. How might we differentiate between these ideas? IMBL, someone's got to say something. I don't know. Yep. So thered be a lot of otherwise fully aquatic air breathing species. We know there are lots and lots of those animals on Earth, and bettas and pet stores are a good example. What else, if we think about a phylogeny, how might we expect those aquatic air breathers to relate to amphibious species? Yeah, and you'd expect a sort of like nested model. That's the first thing I came up with too, so I'm glad we're on the same table. So the first prediction that I wanted to look at was this idea that if the continuum hypothesis is true, we should see amphibious clades nested in air breathing, where the emergence first hypothesis kind of predicts this will happen all over the place. So that basically looks like this. So on the left here, a black line is an aquatic species, a blue one is an air breathing, otherwise aquatic species, and red are amphibious species. And so what I mean by a nested pattern is that first some common ancestor evolved with air breathing ability in water, and then secondarily we see the origin of amphibious natural history. Independence means it's scattered. Some clades will be air-breathing aquatic ones, others will be amphibious, and there won't be a pattern. Any bets? So I based this analysis I'm going to show you on a study by a friend of mine that estimated 87 origins of amphibians. Of those 87 origins, how many do you think occurred in previously air-breathing species? There's ballpark. There's no right or wrong answer. I'm just curious how likely you think this continuum idea is. 80. So a good chunk. 60, good chunk. 7. You saw my slides. 7. 80 of the origins did not come from air breathing. Only 7 did. I still have a hard time believing this, but it's true. Really strong evidence against this continuum idea. I came up with another prediction that I want to show you because I think it's kind of interesting and I'd be interested in what folks here think. The other idea is that the continuum hypothesis predicts that amphibious clades should be older than air breathing. So we all know that evolution doesn't occur according to a plan. There's no like predestined outcome, but still on the whole, if you expect that air breathing has to come first, amphibiousness comes second, that should on average, I would argue, take longer on average than just air breathing evolved. But again, that's not the pattern that we see. So if we take all of these independent origins of amphibiousness versus origins of air breathing in otherwise aquatic fishes, we actually see that the amphibious clades are slightly younger. They're about, on average, these species have been around for about 17 million. So another strike against this continuum hypothesis, which I think is really, really interesting. And it's changed my way of thinking about a lot of these animals and why they're on land. And so the take home from this part is most extant amphibious fishes that we see on Earth probably left water first and then evolved to breathe air. And when I start thinking about some of the common amphibious models, all of a sudden it seems so obvious. And I feel really kind of dumb for not thinking of this earlier. So species like mud skippers and like the killifish I'll talk about earlier, these are really good at breathing air in air. And they have no capacity to breathe air when they're in water. Mud skippers breathe air across highly modified gills. They have no way to expose those gills to air when they're in water. The killifish, as I'll show you in a bit, they breathe air through their skin. And again, there's no way to expose the skin to the air unless they're spending a lot of time jumping, at which point aren't they just kind of semi-terrestrial anyway. So I think that's pretty interesting. And what that means is that our own tetrapod evolutionary history is kind of interesting in the fact that it does not resemble most invasions of land. And so if I spent my PhD year studying killifish to try to understand our own origins, I was barking up the wrong tree completely. What this means is that if we want to understand sort of how we evolved, we probably want to focus on those 7 other clades that were air breathing first, and most of these other species might not be a great model. Does anyone have any questions about this bit before I kind of switch gears? Yeah. 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 Basically. So that's actually what I dug deeper into this data. That's exactly what's going on. Basically, amphibiousness comes from animals that get stranded. Life on land is hard. Fish don't generally. The opportunity on land is almost always not enough to encourage a fish to move on to land. Amphibious evolves in the interclimate. It evolves in places that have really severe dry seasons. That is overwhelmingly, we think, the factor that gives rise to amphibious behavior. Do you think it's more likely for air breathing first and then leaving water? Or the opposite? But when you look at the species level, it's basically the same. It's like roughly 10% of amphibious fish came from aquatic air breathing. And I don't know, maybe that's not surprising to people in this room. That blew my mind. There's really, it didn't exist. That idea didn't really exist in the literature. I think the folks who have studied the invasion of land by fish have always had just such tunnel vision focused on the human story. It's been known for a long time that tetrapods, us, we came from animals that could breathe air and water. And I think that focus on our own sort of evolutionary path just made that the default model that was applied to all sorts of species. That's basically, if you read books about this kind of thing, that's the only way it's presented. And I thought that this was going to just be a no-brainer to support that idea when I did this analysis, to be totally honest. Okay, let's move on. So now we're going to talk about how amphibious fish survive on land. It's hard out there for a fish. How do they do it? And I'm going to tell you some stories in particular about this fish here, the mangrove rivulus. Why the mangrove rivulus? This group down here, the killifishes, the aplocheloids, they did not evolve the ability to breathe air first. They're not good representatives of the tetrapod story. Why do we study those? Basically because the lab had a big colony of them. That's what was available. But the lab had a colony of them because these fish are amazing. And we tried to I spent my whole PhD trying to sort of explain why these fish are superior to other amphibious fishes. And what I've come up with from that sort of years of thought is a scoring system that conveniently shows that killifishes are better than the rest. That was not how we intended this to work out, but it's just so obvious that there's no way around it. So what makes a good amphibious fish? There are a couple ways to do it. So we have some species like the lungfish that just feels intuitively like an amazing example of an amphibious fish that survives on land for years. But then people say, they suck. They can't do anything. They just sit there in a cocoon. Mud skippers, those are the really good amphibious fish. They run around or flop around the mud flat, and they eat on land, they breed on land, they fight each other on land, they do all this stuff. But they actually die if they're out of water for more than a few hours at a time. They always have to go back into water to sort of moisten their surface. They can spend 95% of their overall life on land, but they have to keep dipping back into the water. So we decided to basically split the difference and say, okay, one way you can be a good amphibious fish is to be high on what we call the immersion tolerance axis. So this text might be a bit small, but sort of some species live out of water just for minutes or hours, others weeks, months, and even years. So fish out here, like the lungfish that live for months or years, we put in this level 4 category of really highly amphibious fish. But you can also be really good at doing stuff on land. Lungfish or not, they get a land use score of 1.5. But as I said, the mud skippers up here at the top, they can do all sorts of things. So just intuitively, they're also very highly amphibious fish. So we created this system that takes into account both of these aspects. And as I said, killifish are the best. Here's my favorite amphibious fish, the mangrove rivulus. They can live out of water for months at a time, and they do all sorts of cool stuff when they're on land. Obviously, this is the fish to study. The downside is they're just squiggly little worms. They are this big. They weigh half a gram. They are 2 to 3 centimeters long, an inch long. They're kind of brown, kind of gray, kind of unspectacular. But I'm going to hopefully convince you in the next 20 minutes that they are worth our attention. So these guys live all over the sort of tropical Caribbean, West Atlantic. So northern end of the range is basically the Florida Keys. They're found all through the Caribbean down to northern Brazil, and basically south of the Amazon. And all through that range, they live in habitats that look like this. They live in mangrove swamps, mangrove forests. We're familiar with the mangrove forest, not like the nice coastal. Pretty awesome, right? So there's not much standing water. If you look at this picture, if you look carefully through the roots, you can't see much standing water. A little puddle over here. But this flagging tape indicates where there are crab burrows, and those crab burrows are actually the story. That's where they fit there. This is Pat. She's my PhD supervisor. She's a real keen mangrove explorer. Is a real keen mangrove. So as I said, mangroves are dotted with burrows that are dug by this deep. This is Cardosoma. Cardosoma does this in the mangrove forest floor. It digs these burrows that go down to the water. So even if there's no standing water around, there'll be burrows that are, I don't know, this big. How many fish live in a burrow that's this big? 5, sometimes. 50, yeah, often. It's amazing. They just pack right in there. It's really astounding. So actually, some of the earliest work on mangrove rivulets suggested that they were might be an endangered or threatened species. Because if you drag a seine net through the mangroves, if you put it in a minnow trap, it never ever catch these fish. They were sort of invisible to classic entomology. And then along comes this guy named Scott Taylor, who decided that he could take this cup, he could go to the hardware store and buy a funnel, and he could strap that funnel to the mouth of a cup and jam it in the crab. If you put this cup in the burrow, there's actually no oxygen in that water. The fish are living right in the air-water interface. So you jam the cup in, they swim in through the funnel to get at this sweet, sweet air or this sweet, sweet oxygenated water, and they just pile up in here. So once we had these cup traps, these Taylor traps that we call them now, all of a sudden scientists realized that these rivulets are super abundant in the mangrove. And so we go out there and we catch them, and they immediately start to show us how much they actually dislike the water. We get really nice, clean ocean water, water that they would never have access to in the mangrove, and they still jump right out. So here, these fish are just stuck inside of the bucket, and they'll stay there for days. They'll stay there for a week without jumping back in. These are gambusia and mollies that are similar happening are actually aquatic fish. So that's what it looks like in a bucket. That's kind of how I fell in love with the fish, seeing that behavior in the lab. You go out at night in the mangroves, you can see kind of the same thing. Fish just basking on little roots or twigs above the waterline. First generation GoPro camera. I was so excited to bring this thing to the field, stick it in a crab burrow. This is an old video, but I still have to show it because it blew my mind at the time. This is from 2012. Stuck a GoPro in a crab burrow, and within minutes, well, the crab was molesting the camera because it doesn't want it in its home. But if you see back here, like these fish are just jumping out of the water all the time. So they're associated with the water typically when it's there, but they love living on land here. Crab likes to pose for us. That's actually not the Cardosoma that dug the burrow. That's another crab that just takes advantage of the water source. So one reason they leave water is just, I don't know, for fun. They call it voluntary immersion. Even if water quality is good, like you saw in the bucket, they jump out and they just sit there for days. We have no idea why they do that when conditions are good. Our best guess is it's just this really stereotyped behavior. They're just programmed to jump out. But one of the big questions is why a fish jumps out with no stimuli at all. But as I said earlier, we talked about how disappearing water is kind of one of the major drivers, the major driver for the evolution of amphibious behavior, amphibious physiology, and we certainly see that in the mangrove. So most of the fish are in these crab burrows, but some fish live in ponds. So at our field site in Belize, there's one big pond that looks like this. full of crocodiles, and full of rivulets. But in the dry season, it completely disappears and there's about 3 months where there's no standing water at all. So you see there are coconuts and there are mangrove logs. And those are the key to the success of these fish in the dry season. So this is the crew down in Belize. And here we are actually dissecting a rotting mangrove log. Because these logs are not only home to fish, but first and foremost, they're home to termites and ants. Those termites and ants carve out all sorts of wonderful tunnels in this mangrove wood, and the fish just pile on it. You can get dozens of fish in a log that's sort of this big around and this long. And so you break apart the wood and it kind of looks like this. The fish are just in those. So we think that's the secret to how they survive the dry season. That's how they stay moist and they just hunker down and wait for water. We also think that's why they're found throughout the entire Caribbean. So there are about 1,000 species of killifish. Most of them have ranges on the order of sort of tens to maybe 100 kilometers. These guys are found throughout the whole Caribbean. We think that happens because they go into the woods and then a hurricane comes and just casts them all over the place. So they're on every single mangrove island, basically. So how do they survive for months on end? This is what I tried to understand during my PhD. So we're gonna play another little game. You're gonna tell me the terrestrial challenge. My slides are only in one order, so we're gonna try to order this in terms of what you think the most acute challenge will be to an amphibious fish versus a longer term challenge. So if a fish comes out of water, what's gonna kill it in the first sort of minutes to hours? Desiccation, absolutely. Dehydration, something they're going to lose their body water. So that is true for most fishes. The way that killifish solve that problem is kind of twofold. So first of all, they thicken their skin. So this is a histological cross section of the skin on top of one of these killifish. Here's one in water. Here's one in air. You can see the epidermis gets about 50% thicker. And we've actually done experiments with radioactive water. We've tracked how it moves through the fish. And this thickening actually enables them to hold on to way more water. They might also have some cool stuff going on in their ventral surface that helps them suck it out of the wood. We don't fully understand that, but something interesting is happening. We also talked about the gills. How do they stop the gills from drying out? Most fish, including these guys in water, they breathe through their gills. On land, they seem to just abandon them completely. These gills are a vulnerability. They could get damaged. It could lead to water loss because of all the surface area. So they cover them up. They use this phenomenon called gill remodeling. So for those of you who aren't oriented with gill anatomy, which I assume is most of you, down the middle of this image here is the gill filament. Branching off either side of the filament are the lamellae. These structures here, the lamellae are the site of exchange between a fish and the surrounding water. They're full of blood. And the water passes through these water channels here. And so in a fish living in water has relatively well-defined water channels and the gills are highly functional. But in air, this group of cells that we call the intralamellar cell mass, the least creative name ever, this massive cells enlarges, covers up the gills, reduces surface area. We think it supports them and protects them from damage, but also really importantly, it prevents evaporative water loss. Okay, what comes next? They've solved the dehydration problem. They're holding on to their internal body, water. What's going to kill them next? What's going to kill them sort of on the order of 10 to 15 minutes? Predators, absolutely. We're not going to talk about predators. We're going to talk about physiology, but predators can kill them at any point in this whole process. I don't think timeline matters that much. Gas exchange, wonderful. So how do these guys breathe air? I already kind of told you about it, so I'll just show you some pretty pictures. So they abandon their gills, they start breathing through the skin. So this is a technique called immunohistochemistry. So what we've done is we've taken cross sections through the skin of fish and we've stained them with an antibody that targets endothelial cells. So these are capillaries and really small blood vessels. So this is a fish in water. Can't see much going on. That's kind of the point. The skin surface is right here. And there aren't a lot of capillaries. even after just 1 day in air, we see this massive, massive angiogenesis. So this structure here is actually a scale. And you can see the blood vessels kind of penetrating between adjacent scales and forming this really interesting network of capillaries right on the skin surface. It's within 1 or 2 microns of the air, one of the thinnest diffusion distances that we know of in any fish. And then after 3 days in air, we see even more formation of these cutaneous blood vessels. And they're able to get all of their oxygen and excrete all of their carbon dioxide through the skin in this one. Okay, what comes next? You can breathe and you can hold on to your water. What's going to kill you on the order of like days to a week? Starvation. Starvation, we'll get to that, but they can survive weeks, no problem. Something else might kill them first. Predators can kill them at any time. We're going to just say predators are this big sort of Damocles hanging over amphibious fish all the time. Yeah. Locomotion is a really important challenge for these fish to solve, but let's think about a rivulus packed into a log. They've solved it behaviorally in terms of choosing a habitat that doesn't require locomotion. Excretion of bingo. I think that's the next challenge, is getting rid of nitrogenous waste. So we get rid of this as urea. So most fish, I'll back up, most fish get rid of nitrogen. Nitrogen waste comes from breaking down protein. Most fish get rid of nitrogenous waste as ammonia, which just diffuses into the water over the gills. But gills of these fish are non-functional. What we do as humans is we convert the nitrogen, the ammonia, into urea, which takes energy. It's expensive to do that, but urea is much less toxic than ammonia, so we can let it build up and we can excrete it using not very much water. So we actually tested the hypothesis that these killifish get rid of nitrogenous waste using urea production. Lots of amphibious fish do that, but they don't. What else might they do? You have an idea, or that's in the stretch? How else can you get rid of nitrogen? You can make, yeah, like be a bird. These fish are not birds, unfortunately. You can dilute it, but that takes a lot of water, and water is really scarce for these fish, so they can't do it. Yeah, they fart it out over the skin. It's amazing. So there's one group of amphibious fish that excretes gaseous ammonia into their gas bladder, into their lung, and then they basically burp it out. But what these guys do is they just get rid of it through those dense capillary networks on the skin. We've done a lot of work trying to understand how that process actually takes place, how it works. I won't bore you with all the details, but I want to show you one pretty picture of the skin. So here again is the epidermal layer, and we've used this immunohistochemistry technique again to label 2 proteins that we think are really important. So the first in green, I apologize to the colorblind in the audience, but I did use red and green. I'm not on the ball. Anyway, the green, which is right here in this crypt of this cell, is an RH protein. This is a channel that allows ammonia to move through. Surrounding that channel is another protein called sodium potassium ATPase, which moves sodium potassium. And so what this does is it allows ammonia to move across the skin. So in the body, ammonia is in an ionic form, so it's actually ammonium, NH4+. Comes to the skin surface, moves through this RH channel, and sits on the skin surface. But it's this ionic form that stays in the mucus or in the boundary layer. The next step is to turn it into ammonia gas so it can fly away and this concentration gradient can persist. So the sodium potassium ATPase actually works really, really hard to make the skin surface more alkaline. Increase the pH, think back to basic chemistry. If you increase the pH, you're getting rid of hydrogen ions. So it's less likely for ammonia to be in this. ionic form, more likely to be in the gaseous form, and it flows away. So they're just pumping really hard to keep their skin surface quite basic, and they're just farting out all their nitrogen. Okay, what's next? It's been said already, so maybe I'll cut to the chase. Sort of weeks to months, we think feeding becomes really, really important. Fish can survive a long time without food, but not forever. I also had to put in this graph because IMBL is doing a lot of respirometry, and I want to show you that my life is respirometry. And it gets old fast, but it's a useful technique. So here on this graph, I'm going to show you the oxygen consumption, which is the proxy of metabolic rate on the y-axis for fish that are in water or that have been air exposed for 3 weeks. As over the first 7 days, metabolic rate is constant because these fish are doing all sorts of stuff. Feeding becomes a challenge longer term. And what we see is metabolic rate drops quite a bit by sort of like 50 to 70%. They're not lungfish, they're not dropping 95%. But metabolic depression seems to conserve the onboard energy and gives them weeks to months of extra survival time on land. What else? Longer term. We talked about fish having to move around, fish having to withstand gravity. That's what I'm going to end with. So the last terrestrial challenge is one that's near and dear to me. This is something I thought about a lot during my PhD. How do amphibious fish deal with gravity when they're on land? And this idea started percolating. This is actually how I started working with Doug all these years ago, as we started thinking about the mechanical properties of fish gills and fish bones. And I read the literature at the time, and there was this idea that the ability to sense body weight and change to respond to gravity was a tetrapod trait. The idea was fish live in water, they're weightless. They don't need to sense body weight. That's something that happened when tetrapods marched onto land. But as I said, tetrapods are one of only many, many origins of amphibious behavior. What happens in these amphibious killifish? Can they actually sense gravity? So the first thing I did was I stretched the gills of these fish. It was hard. These fish, as I said, weigh half a gram. The gills are like a millimeter long. So I dissect them out, I pull on them, and after the fish were on land for a couple of weeks, the bones in their gill arches actually got quite a bit stiffer, which was really exciting news. It suggested perhaps that these fish are responding to gravity. We talk a lot in IMBL about critical and persuasive prediction. And would you call this result critical or persuasive? The fact that an air exposed fish stiffens its gill, does that mean it's responding to gravity? No, there's all sorts of things that happen when a fish is on land. How do we know that this is due to gravity? How can we test that? I visited some friends at Dalhousie University in Halifax who are the proud owners of something we call a random positioning machine. There are like 5 of these on the planet. They're very fun. We load fish into them. The fish are so small, they live in 6-well plates. No problem at all. Load them into 6-well plates, and then they spin around for a week. And they're fine. There's a bit of cotton balls in there, so it's moist. And they spin around, and this simulates zero gravity by averaging out the force over time. So if you think about an animal that's right side up half the time, upside down half the time, in that one plane, gravity is going to average out to 0. By spinning them all over the place, on every plane, gravity averages out to 0. So that's what I did. And then I measured gill stiffness, and the stiffening went away. You could have an air exposed fish without gravity, and the gills were just like the gills of a fish in water. So I take that as pretty good evidence that these fish are actually sensing changes in gravity. We're getting to 45 minutes, so I'm going to blast through this part, but I was really curious how this happens. So I did some electron microscopy. I zoomed right in on the bone of these fish, all the way in so we can see an osteoblast here. This is the cell that actually creates bone. Down here is the bone itself. And between these two, these are actually individual collagen molecules, little fibers that are cut right through. They look like little dots. After fish were air exposed, you can see that this collagen is a lot more densely packed, and we start to see this dark mineral layer. So we think these are sort of the keys to stiffening a bone. You add more calcium, you pack your collagen right in there, the bone gets stiffer. We also did some proteomics to think about all the other proteins that are changing. So this is a heat map that in red, it shows you proteins that are increased in expression in air exposed fish. Again, cut to the chase. This list of proteins was the easiest thing ever to interpret because every protein that changes abundance in these amphibious fish is known from human study. These are all the same proteins that are associated with bone growth in humans and other mammals. So not only are these fish sensing gravity just like us, but they respond using the same mechanisms that we do. So we now think that sensing gravity is probably not at all a tetrapod trait. It's probably goes back to the origin of vertebrates. So to wrap up my whole PhD in this whole section in one sentence, to be amphibious really requires the ability to change your physiology. We call this physiological plasticity. If you're going to move between very different environments, you can't be the same fish. You have to be able to change how you interact with the environment. So I want you to leave this room tonight with basically two key takeaways. So first of all, there are many ways for fish to move from water to land. This is something that happens again and again and again. And what we now think is that air breathing comes first only in a very few cases. Typically, fish lose water. They're forced into this really, really harsh environment, and they've got to figure it out. Secondly, life on land is hard. Amphibious fishes must overcome all sorts of challenges, and they do so basically in a diversity of ways that mirrors the diversity of species on the planet. I showed you the strategies used by killifish. We could zoom in on another taxon like a mud skipper or a lungfish. And unfortunately, or maybe fortunately, it's pretty awesome. You would get kind of a whole different list of answers. There's so many ways to be an amphibious fish. It's just kind of astounding. But I'm out of time, so I'm just going to say thank you to the people I've been able to work with. Thank you for listening, and I'd love to take questions.
Excellent talk, Andy. Thank you very much. Questions, folks? Everyone's mind is blown about it.
I'm still a little bit confused on in what situation has anyone looked at what situations like those structural supports come before the air breathing, like in amphibious fishes?
You're talking about things like really strong pectoral fins to walk around.
You said there was the continuum theory and the alternative to that, which you didn't really seem to like very much.
Well, I like them all. I just want to know what actually explains life on Earth. So you're interested in how the ability to walk underwater might serve as a pre-adaptation for life on Earth. That certainly seems to be the case for our ancestors. Like early tetrapods were probably these creatures that lived in marshes or reed beds or that kind of thing, where it was really hard to swim, probably very low dissolved oxygen in the environment, and so they evolved pectoral fins that let them sort of push their way through this dense vegetation. They evolved the ability to breathe air, so they didn't need to rely on aquatic oxygen, and then they were basically set up for life on land. But that doesn't seem to have happened that many times. There's so many fish that don't swim, benthic fish that have found ways to sort of walk and crawl around. Not many of those. Well, I mean, mud skippers have done it, polypterus has done it, but on the whole, most amphibious fish aren't very good at that kind of locomotion. So I think, again, sadly, there's not a lot of broad sort of commonality. That explains some origins of amphibious behavior, but not most of them.
We were wondering, you talked quite a bit about moving from the water to land and some of those physiological changes and the time that they take, a couple days for the capillaries and the bone structure to change. What about going the other way? Like, is it the same time frame when they move from land back into water?
So we don't know for a lot of traits, but it seems basically everything seems to be reversible. For the traits we've looked at in detail, the transition back to water seems to be a bit slower. And that makes intuitive sense to us because it's really urgent to remodel your structures when you move on to land because the consequence is death in many cases. Whereas when you're moving the other way, like there's still trade-offs. So these like dense capillary networks in the skin probably have some drawbacks for fish and water. And fish might actually lose oxygen to the environment through those capillaries and across the skin. They might increase optimal regulatory demands, make them more prone to injury. But it's probably not a life or death thing, so it seems to reverse a bit more slowly. So gills, for example, the gill remodeling occurs in a few days, takes about a week to be reversed. The structural changes to the gill bones occur in 1 to 2 weeks. They take 16 weeks to be reversed. So it seems to be more like the reason that the fish don't have things like strong gills is basically because why spend energy on it? Move on to land, it's worth investing in a stronger gill, but there's not a big drawback. You don't get rid of it right away. The 16-week turnaround is actually kind of interesting because it matches up really well with what we know about just general bone turnover in those fish. So basically the bone is constantly being remodeled. And as it gets remodeled, if they're living in water, it just gets built back into the aquatic. But there's not this urgency to reduce it.
Hey, um, how it looked like in the pictures you showed us, they had fairly relatively large eyes. How do have their sensory, how adapted sensorily to terrestrial life?
Know how well they can see where they live. It's kind of a puzzle, and one of my colleagues is working on that right now. The big eyes are kind of so basically you can have an eye just like when you go underwater, the world gets blurry. Fish can basically have an eye that is well suited to seeing in air or in water. Some weirdos like the four-eyed fish have solved that problem by having two sets. But for most fish, unless you can really dynamically change the properties of your eyes, you're built one way or the other. The intermediate option to me seems like it would be just the worst of both worlds. We've been really curious for years about what the killifish can see, but they're so small that none of the classic techniques work. They don't seem to be able to see very well on land, but the water they live in is really murky and crappy. So I don't know why they would need to have good eyes for seeing in water. It's a bit of a puzzle. They hunt in both environments. So they're very voracious predators in water. They sort of sit at the top of the crab burrows and wait for something to fall in and they grab it. But we also know that they forage on land. So they can cover tens of meters in about 15 minutes, just flipping around the forest floor. While they're doing that, they can grab a termite in their jaws, but then they're stuck. They can't swallow it. It's very funny. They're suction feeders, like basically all fish, and they have no way to get it down without the dynamic flow of water. So they'll hold on to that termite. They can grab it in their jaws, and they have to drag it to a crab burrow, go in the water, and then they can process it. Hilarious. But that doesn't answer your question about where they can see. We just don't know.
So you mentioned that in killifish, it's likely that the ability to breathe is secondary. Do you think that like they kind of were in the water, like in the ocean, and then they got stranded in the mangroves by a hurricane or something?
Where they came from, it's likely that they were freshwater first and migrated towards the coast. The vast majority of rivulet species anyway are freshwater species. but they're all specialists in puddles and ephemeral streams and that kind of thing, but does that answer your question?
It would make sense for why they don't like seawater.
Well, so those guys love seawater. Well, I mean, so the fish I studied, this is another amazing, the list of amazing facts about them just keeps going on. So they have one, they're super euryhaling. So it means they can take whatever salinity you throw at. You can take one of those fish and put it in reverse osmosis water. And water with no ions. It kills most fish. They're fine. You can take a fish from brackish water, throw it right in. It's fine. The only issue is a buoyancy issue because the density of the water changes and they have to adjust the swim bladder. It's pretty funny. You can go the other way. You can take that fish and you can put it in 120 parts per thousand. So like 4 times seawater. Again, straight up, and they're fine. They just float for a long time until they figure out how to adjust the buoyancy and think. So salinity doesn't bother them at all, but a lot of the other jellyfish are. Then you have the fungulates, like there are amphibious fish out here at Shoals, but a totally different group of killifish, a totally independent, we think, origin of amphibious behavior. I could talk about them for another hour. They're awesome.
With the killifish, when dealing with the nitrogenous waste, when they're changing the ammonia to NH3, are they able to withstand the acidic conditions, or does that not really affect them? Because I assume in that situation they're still in like the tree, the mangrove roots, so wouldn't the acidification start to affect?
That's a very good question. If they were on a really acidic substrate, that might pose a big challenge. I feel like they could solve this problem behaviorally as long as they can get part of their skin off the substrate and expose it to air. Maybe they could shunt blood and therefore ammonia to that region. I don't know. I've never really thought about that, but yeah, acid would be placed down in the color.
Are there habitats that are more common for amphibious fish just in general, like that, like out of those 87 that are there, is there like distribution that are like most common in certain area? Or not certain area, but certain like type of habitat?
So they're largely tropical. The fungulates that live here are one of the very few examples of a temperate amphibious fish. And then the other big sort of broad pattern is they live in marginal conditions in places where the water doesn't persist all year. So usually shallow, either like shallow inland swamps and stuff, especially in parts of the world that have really strong wet and dry seasons, or the intertidal. I guess in the temperate intertidal, there are quite a few. But there are also rock animals that live out here that are pretty amphibious. But the bulk are tropical. There is a lab in Portugal that has been very interested in the epigenetics of these fish. And like I said, I could tell you about the weird things about these fish for ages. The other really remarkable thing about them that I don't know how I haven't said yet, they are the world's only self-fertilizing hermaphroditic vertebrate. So between 90 and 99% of these animals in nature have functional ovaries and testes that are all kind of mixed together. And they have internal fertilization, and we call it an isogenic embryo pops out. So they're super inbred. You lose half of your heterozygosity with every generation of inbreeding, basically. And we know in the wild they can inbreed for dozens of generations. We'll find a pond with hundreds of animals that are all basically clones of one another. So basically, the heritable aspect of their lives is epigenetic. So there's quite strong epigenetic inheritance of all sorts of different traits, but I'm not the one who's done that work, so I can't tell you much about it. Good dodge, huh?
Yeah. Not reading this before, yeah, I'm sure you do now. So thank you very much, Andy. Awesome.